Bats are mammals of the order Chiroptera whose forelimbs form webbed wings, making them the only mammals naturally capable of true and sustained flight. By contrast, other mammals said to fly, such as flying squirrels, gliding possums, and colugos, can only glide for short distances. Bats do not flap their entire forelimbs, as birds do, but instead flap the spread-out digits, which are very long and covered with a thin membrane of patagium. Bats are the second largest order of mammals, representing about 20% of all classified mammal species worldwide, with about 1,240 bat species divided into two suborders: the less specialized and largely fruit-eating megabats, or flying foxes, and the highly specialized and echolocating microbats. About 70% of bat species are insectivores. Most of the rest are frugivores, or fruit-eaters. A few species, such as the fish-eating bat, feed from animals other than insects, with the vampire bats being hematophagous, or feeding on blood. Bats are present throughout most of the world, with the exception of extremely cold regions. They perform the vital ecological roles of pollinating flowers and dispersing fruit seeds. Many tropical plant species depend entirely on bats for the distribution of the seeds. Bats are economically important as they consume insect pests, reducing the need for pesticides. The smallest bat is the Kitti's hog-nosed bat, measuring 29 to 34 millimeters in length, 15 centimeters across the wings, and 2 to 2.6 grams in mass. It is also arguably the smallest extant species of mammal, with the Etruscan shrew being the other contender. The largest species of bat are a few species of Pteropus and the giant golden-crowned flying fox, with a weight up to 1.6 kilograms and wingspan up to 1.7 m. Etymology. In many languages, the word for bat is cognate with the word for mouse. For example, chauve-souris in French, murciélago in Spanish, sigurzaha in Basque, in Russian, slijepimis in Bosnian, nakir in Estonian, vlamwi in Afrikaans, from the Dutch word vleermuis. An older English name for bats is flittermouse, which matches the name in other Germanic languages related to fluttering of wings. As for Latin bacchi, a moth nocturnal insect. Classification and evolution. Bats are placental mammals. Bats were formerly thought to have been most closely related to the flying lemurs, tree shrews, and primates, but recent molecular cladistics research indicates that they actually belong to Laurasia, a diverse group also containing carnivora and Artiodactyla. The two traditionally recognized suborders of bats are Megachiroptera, Microchiroptera. Not all megabats are larger than microbats. The major distinctions between the two suborders are: microbats use echolocation, with the exception of the genus Rusatis. Megabats do not. Microbats lack the claw at the second finger of the forelimb. The ears of microbats do not close to form a ring. The edges are separated from each other at the base of the ear. Microbats lack underfur. They are either naked or have guard hairs. Megabats eat fruit, nectar, or pollen. Most microbats eat insects. Others may feed on fruit, nectar, pollen, fish, frogs, small mammals, or the blood of animals. Megabats have well-developed visual cortices and show good visual acuity, while microbats rely on echolocation for navigation and finding prey. The phylogenetic relationships of the different groups of bats have been the subject of much debate. The traditional subdivision between Megachiroptera and Microchiroptera reflects the view that these groups of bats have evolved independently of each other for a long time, from a common ancestor already capable of flight. This hypothesis recognized differences between microbats and megabats and acknowledged that flight has only evolved once in mammals. Most molecular biological evidence supports the view that bats form a single or monophyletic group, Researchers have proposed alternative views of Chiropteran phylogeny and classification, but more research is needed. In the 1980s, a hypothesis based on morphological evidence was offered that stated the Chiroptera evolved flight separately from the Chiroptera. The so-called flying primate hypothesis proposes that when adaptations to flight are removed, the Chiroptera are allied to primates by anatomical features not shared with Chiroptera. One example is that the brains of megabats show a number of advanced characteristics that link them to primates. Although recent genetic studies strongly support the monophyly of bats, debate continues as to the meaning of available genetic and morphological evidence. Genetic evidence indicates that megabats originated during the early Eocene and should be placed within the 4 major lines of microbats. Consequently, 2 new suborders based on molecular data have been proposed. The new suborder of Chiroptera includes the Pteropodidae, or megabat family, as well as the families Rhinolophidae, Hipposidridae, Crasionecteridae, Megadermatidae, and Rhinopermatidae. The other new suborder, Chiroptera, includes all of the remaining families of bats. These 2 new suborders are strongly supported by statistical tests. Teeling found 100% bootstrap support in all maximum likelihood analyses for the division of Chiroptera into these 2 modified suborders. This conclusion is further supported by a 15-base pair deletion in BRCA1 and a 7-base pair deletion in PLCB4 present in all Chiroptera and absent in all Chiroptera. Perhaps most convincingly, a phylogenomic study by Zarkov-Georg et al. Showed that the 2 new proposed suborders were supported by analyses of thousands of genes. The Chiropteran phylogeny based on molecular evidence is controversial because microbat paraphyly implies that one of 2 seemingly unlikely hypotheses occurred. The first suggests that laryngeal echolocation evolved twice in Chiroptera, once in Chiroptera and once in the rhinolophoids. The second proposes that laryngeal echolocation had a single origin in Chiroptera, was subsequently lost in the family Pteropodidae, and later evolved as a system of tongue-clicking in the genus Rusitus. Analyses of the sequence of the vocalization gene, FOX2, were inconclusive as to whether laryngeal echolocation was secondarily lost in the pteropodids or independently gained in the echolocating lineages. However, analyses of the hearing gene, Preston, seem to favor the independent gain in echolocating species rather than a secondary loss in the pteropodids. In addition to Chiroptera and Chiroptera, the names Pteropodiformes and Vespertilioniformes have also been proposed for these suborders. Under this new proposed nomenclature, the suborder Pteropodiformes includes all extant bat families more closely related to the genus Pteropus than the genus Vespertilio, while the suborder Vespertilioniformes includes all extant bat families more closely related to the genus Vespertilio than to the genus Pteropus. Little fossil evidence is available to help map the evolution of bats, since the small, delicate skeletons do not fossilize very well. However, a late Cretaceous tooth from South America resembles that of an early Microchiropteran bat. Most of the oldest known, definitely identified bat fossils were already very similar to modern microbats. These fossils, Icaronecterized, Archaeonecterized, Paleochiropteryx and Herceonecterized, are from the early Eocene. Eocene period, 52.5 million years ago. Archaeopteropus, formerly classified as the earliest known megachiropteran, is now classified as a microchiropteran. Bats were formerly grouped in the superorder Archonta, along with the tree shrews, colugos, and the primates. Because of the apparent similarities between megachiroptera and such mammals, genetic studies have now placed bats in the superorder Laurasiatheria, along with carnivorans, pangolins, odd-toed ungulates, even-toed ungulates, and cetaceans. A recent study by Zhang et al. Places Chiroptera as a sister taxon to the clade Perissodactyla. However, the first phylogenomic analysis of bats shows that they are not sisters to Perissodactyla. Instead, they are sisters to a larger group that includes ungulates and carnivores. The traditional classification of bats is order Chiroptera, suborder Megachiroptera, Pteropodidae, suborder Microchiroptera, superfamily Emballoneuroidea, Emballoneuroidae, superfamily Mollussoidea, Mollussoidae, superfamily Nataloidea, Furipteridae, Mysopodidae, Natalidae, Thyropteridae, superfamily Noctilionoidea, Mormupodimus, Noctilionide, Philostomodidae, superfamily Rhinolophoidea, Megadermatidae, Nycteridae, Rhinolophidae, Hipposidridae, superfamily Rhinopomatoidea, Crassionycteridae, Rhinopomatidae, superfamily Vespertilionoidea, Vespertilionidae, Antrozoidae. Megabats primarily eat fruit and nectar. In New Guinea, they are likely to have evolved for some time in the absence of microbats, which has resulted in some smaller megabats of the genus Nyctamine becoming insectivorous to fill the vacant microbat ecological niche. Furthermore, some evidence indicates that the fruit bat genus Deralipex from the Solomon Islands, and its close relative Miramiri from Fiji, have evolved to fill some niches that were opened because there are no non-volant and non-flying mammals on those islands. Fossil bats. Fossilized remains of bats are few, as they are terrestrial and light-boned. Only an estimated 12% of the bat fossil record is complete at the genus level. Fossil remains of an Eocene bat, Echolocating, were found in 1960. Another Eocene bat, Echolocating finni, was found in the 52-million-year-old Green River Formation in Wyoming, United States, in 2003. This intermediate fossil has helped to resolve a long-standing disagreement regarding whether flight or echolocation developed first in bats. The shape of the ribcage, faceted infraspires fossa of the scapula, manus morphology, robust clavicle, and keeled sternum all indicated Echolocating was capable of powered flight. However, the well-preserved skeleton showed that the small cochlea of the inner ear did not have the morphology necessary to echolocate. Finni lacked an enlarged orbital apophysis on the malleus, and a stylohyle element with an expanded paddle-like cranial tip, both of which are characteristics linked to echolocation in other prehistoric and extant bat species. Because of these absences, and the presence of characteristics necessarily for flight, Echolocating provides strong support for the flight-first hypothesis in the evolution of flight and echolocation in bats. The appearance and flight movement of bats 52.5 million years ago were different from those of bats today. Echolocating had claws on all 5 of its fingers, whereas modern bats have at most 2 claws appearing on 2 digits of each hand. It also had longer hind legs and shorter forearms, similar to climbing mammals that hang under branches, such as sloths and gibbons. This palm-sized bat had short, broad wings, suggesting that it could not fly as fast or as far as later bat species. Instead of flapping its wings continuously while flying, Echolocating likely alternated between flaps and glides while in the air. Such physical characteristics characteristics suggest that this bat did not fly as much as modern bats do, rather flying from tree to tree and spending most of its waking day climbing or hanging on the branches of trees. The distinctive features noted on the Onychodonectari's fossil also support the claim that mammalian flight most likely evolved in arboreal gliders, rather than terrestrial runners. This model of flight development, commonly known as the trees down theory, implies that bats attained powered flight by taking advantage of height and gravity, rather than relying on running speeds fast enough for a ground-level takeoff. The mid-Eocene genus Necromantis is one of the earliest examples of bats specialized to hunt vertebrate prey, as well as one of the largest bats of its epoch. The late Eocene Witwacia is another similarly large predatory bat, while Egyptonectaris is among the first and largest omnivorous bat species. The extinct bats Palaeochiropteryx tupiodin and Acienectaris kumari are the first fossil mammals to have the coloration discovered, both of a reddish-brown habitats. Flight has enabled bats to become one of the most widely distributed groups of mammals. Apart from the Arctic, the Antarctic and a few isolated oceanic islands, bats exist all over the world. Bats are found in almost every habitat available on Earth. Different species select different habitats during different seasons, ranging from seasides to mountains and even deserts. But bat habitats have two basic requirements: roosts, where they spend the day, or hibernate, and places for foraging. Most temperate species additionally need a relatively warm hibernation shelter. Bat roosts can be found in hollows, crevice, foliage, and even human-made structures, and include tents the bats construct by biting leaves. The United States is home to an estimated 45 to 48 species of bats. The three most common species are Myotis lucifugus, Heptizocus fuscus, and Tadyrida brasiliensis. The little and the big brown bats are common throughout the northern two-thirds of the country, while the Mexican free-tailed bat is the most common species in the southwest, sometimes even appearing in portions of the southeast. Anatomy. Wings. The finger bones of bats are much more flexible than those of other mammals, owing to their flat and cross-section and to low levels of minerals, such as calcium, near the tips. In 2006, Sears et al. Published a study that traces the elongation of manual bat digits, a key feature required for wing development, to the upregulation of bone morphogenetic proteins. During embryonic development, the gene controlling BMP signaling, BMP2, is subjected to increased expression in bat forelimbs, resulting in the extension of the offspring's manual digits. This crucial genetic alteration helps create the specialized limbs required for volant locomotion. Sears et al. Also studied the relative proportion of bat forelimb digits from several extant species and compared these with a fossil of Echolocating index, an early extinct species from approximately 50 million years ago. The study found no significant differences in relative digit proportion, suggesting that bat wing morphology has been conserved for over 50 million years. The wings of bats are much thinner and consist of more bones than the wings of birds, allowing bats to maneuver more accurately than the latter, and fly with more lift and less drag. By folding the wings in toward the bodies on the upstroke, they save 35% energy during flight. The membranes are also delicate, ripping easily. However, the tissue of the bat's membrane is able to regrow, such that small tears can heal quickly. The surface of the wings is equipped with touch-sensitive receptors on small bumps called Merkel cells, also found on human fingertips. These sensitive areas are different in bats, as each bump has a tiny hair in the center, making it even more sensitive and allowing the bat to detect and collect information about the air flowing over its wings, and to fly more efficiently by changing the shape of its wings in response. An additional kind of receptor cell is found in the wing membrane of species that use the wings to catch prey. This receptor cell is sensitive to the stretching of the membrane. The cells are concentrated in areas of the membrane where insects hit the wings when the bats capture them. The teeth of microbats resemble insectivorans. They are very sharp to bite through the hardened armor of insects of the skin of fruit. Mammals have one-way valves in the veins to prevent the blood from flowing backwards, but bats also have one-way valves in their arteries. The tube-lipped nectar bat has the longest tongue of any mammal relative to its body size. This is beneficial to them in terms of pollination and feeding. The long, narrow tongues can reach deep into the long cup shape of some flowers. When the tongue retracts, it coils up inside its rib cage. Bats possess highly adapted lung systems to cope with the pressures of hard flight. Flight is an energetically taxing aerobic activity and requires large amounts of oxygen to be sustained. In bats, the relative alveolar surface area and pulmonary capillary blood volume is significantly larger than most other small quadrupedal mammals. Echolocation. Bat echolocation is a perceptual system where ultrasonic sounds are emitted specifically to produce echoes. By comparing the outgoing pulse with the returning echoes, the brain and auditory nervous system can produce detailed images of the bat's surroundings. This allows bats to detect, localize, and even classify the prey in complete darkness. At 130 decibels in intensity, bat calls are some of the most intense airborne animal sounds. To clearly distinguish returning information, bats must be able to separate their calls from the echoes that they receive. Microbats use two distinct approaches. Low-duty cycle echolocation. Bats can separate their calls and returning echoes by time. Bats that use this approach time their short calls to finish before echoes return. This is important because these bats contract the middle ear muscles when emitting a call, so they can avoid deafening themselves. The time interval between the call and echo allows them to relax these muscles, so they can clearly hear the returning echo. The delay of the returning echoes provides the bat with the ability to estimate the range to the prey. High-duty cycle echolocation. Bats emit continuous call and separate pulse and echo in frequency. The ears of these bats are sharply tuned to a specific frequency range. They emit calls outside of this range to avoid self-deafening. They then receive echoes back at the finely tuned frequency range by taking advantage of the Doppler shift of the motion in flight. The Doppler shift of the returning echoes yields information relating to the motion and location of the bat's prey. These bats must deal with changes in the Doppler shift due to changes in the flight speed. They have adapted to change the pulse emission frequency in relation to the flight speed so echoes still return in the optimal hearing range. The new Chiroptera and Chiroptera classification of bats, supported by molecular evidence, suggests two possibilities for the evolution of echolocation. It may have been gained once in a common ancestor of all bats and was then subsequently lost in the Old World fruit bats, only to be regained in the horseshoe bats, or echolocation evolved independently in both the Chiroptera and Chiroptera lineages. Two groups of moths exploit a bat's sense to echolocate. Tiger moths produce ultrasonic signals to warn the bats that they are chemically protected or aposematic. Other moth species produce signals to jam bat echolocation. Many moth species have a hearing organ called a tympanum, which responds to an incoming bat signal by causing the moth's flight muscles to twitch erratically, sending the moth into random evasive maneuvers. In addition to echolocating prey, bat ears are sensitive to the fluttering of moth wings, the sounds produced by timberlate insects, and the movement of ground-dwelling prey, such as centipedes, earwigs, etc. The complex geometry of ridges on the inner surface of bat ears helps to sharply focus not only echolocation signals, but also to passively listen for any other sound produced by the prey. These ridges can be regarded as the acoustic equivalent of a Fresnel lens, and may be seen in a large variety of unrelated animals, such as the lesiga, bat-eared fox, mouse lemur, and others. By repeated scanning, bats can mentally construct an accurate image of the environment in which they are moving and of the prey item. Other senses. Although the eyes of most microbat species are small and poorly developed, leading to poor visual acuity, no species is blind. Microbats use vision to navigate, especially for long distances when beyond the range of echolocation, and species that are gleaners, that is, ones that attempt to swoop down from above to ambush insects, like crickets on the ground or moths up a tree, often have eyesight about as good as a rat's. Some species have been shown to be able to detect ultraviolet light, and most cave-dwelling species have developed the ability to utilize very dim light. They also have high-quality senses of smell and hearing. Bats hunt at night, reducing competition with birds, minimizing contact with certain predators, and travel large distances in the search for food. Megabat species often have excellent eyesight, as good as, if not better than, human vision. This eyesight is, unlike its microbat relations, adapted to both night and daylight vision and enables the bat to have some color vision, whereas the microbat sees in blurred shades of gray.